bright lights. Ooh, just pull the chair over maybe.
I use pre-type to say.
All right, we will have people coming in late because there are classes ending and they can't get here on time. And we have, and we'll accommodate folks without difficulty. But I wanted to get started on time, and representing the Winslow Medal Committee, its chair, Professor Melinda Irwin, and the nominator, Professor Vasilis Vasiliou. It's my great pleasure to welcome all of you to the 7th conferment of the CEA Winslow Medal to Dr. Linda Birnbaum. The Winslow Medal is given in honor of the founder of our school, and many would say the individual who truly conceptualized the modern state public health department in the United States. And the gentleman is over to your left. The school's medal is our highest honor in our school. It's conferred every few years, not every year. And it is designed to be given to world-renowned innovators with distinguished careers and outstanding leadership in public health, whether it's scholarship, education, contributions to society. Past recipients have included many luminaries in our field, but including Sir Richard Doll, Dr. Judith Rodin, and Dr. Anthony Fauci, and others who have really contributed immeasurably to public and global health. Before introducing Dr. Birnbaum, I wanted to say a few words about CEA Winslow. He was born in 1877, received his bachelor's and master's degrees from MIT in 1898 and 1899, respectively. He taught at the University of Chicago, City College, Columbia, and Yale, and served as the editor of the Journal of Bacteriology, and also he was editor of the American Journal of Public Health. He was president, in fact, of the American Public Health Association, and the APHA uses Winslow's definition of public health as its own definition of public health on its website to this day. At Yale, Professor Winslow was chair of the Department of Public Health at its inception in 1950. During his 30-year tenure here, he brought Yale, the department, and himself considerable international and national distinction and acclaim. During this period of time, the field of hygiene began to encompass preventive medicine, a somewhat broader theme, and bacteriology evolved to include parasitology, mycology, and virology. A classic epidemiology evolved to include clinical epidemiology. The control of communicable diseases evolved to include the chronic and acute non-communicable diseases. And public health was the heart of a focus on social determinants of illness and health equity. And as I mull this over, one thinks of the French phrase, plus que ça change, plus que c'est la même chose. You know, the more things change, the more things stay the same, because we seem to have to reinvent these insights of Winslow and his colleagues from 100 years ago, even today. So Winslow developed public health into a premier educational program that graduated not only students with master's and doctoral degrees in public health, but also medical students imbued with a preventive spirit. Yale was the very first academic institution in the United States to grant a degree in public health. And in 1946, we were one of 6 of the very first schools to be accredited for degree granting in public health. Many of the advances made in public health nationally and internationally during the latter half of the last century were made by those who followed Winslow at Yale. These included John Rodman Paul, Dorothy Horseman, Jim Niederman, John Thompson, Max Thieler, Jordi Casals, And I thought of adding people like Nancy Ruddle, but then I realized they're still in our faculty, so I thought I wouldn't add them. But really, folks who built on the intellectual vision of Winslow. Now, the research achievements of our faculty, staff, and our many graduates continues to move our field towards prevention and alleviation of disease, disability, and suffering. And for that, I am very proud. This year's Winslow Medal recipient, Dr. Linda Birnbaum, is Scientist Emerita and former director of the NIEHS, but for those of you who are not aficionados, the National Institute of Environmental Health Sciences at the NIH. It's a little unusual because it's located in the triangle area of North Carolina, unlike the other institutes that have sort of a Bethesda Rockville base. Her contributions continue in her current role as scholar in residence at Duke University's Nicholas School for the Environment. The Yale School of Public Health has selected Dr. Birnbaum for its Winslow Medal to honor her foundational work in 3 areas: defining the health hazards of persistent organic pollutants, uncovering the mechanisms of action of toxic environmental contaminants, and linking real-world exposures to human health problems. She was the first toxicologist and the first woman to lead the NIEHS, and she led the paradigm development that nothing is just genetics and nothing is just environment, but all health is an integration of both genes and the environment. Dr. Birnbaum demonstrated that early life contaminant exposures were associated with adverse health effects in children. She continues to provide national and international leadership in the area of PFAS, these forever chemicals that have been demonstrated have multiple harmful properties to humans, and developed a vigorous program in community engagement and involvement in environmental health studies. And she was a pioneer in this area. In other words, community consultation was not necessarily what the NIH was good at when Dr. Birnbaum started her work at NIEHS. She's led the Toxicology Division of the American Society of Pharmacology and Experimental Therapeutics, the American Aging Society, the Society of Toxicology, the International Union of Toxicology, leadership positions in all those organizations, and is an elected member of the National Academy of Medicine. As the NIEHS director from 2009 to 2019, Dr. Birnbaum also directed the National Toxicology Program. She oversaw a budget of more than $740 million. She led the funding of over 1,000 research grants to discover how the environment influences human health and disease. And this institute also, of course, supports substantial training programs, educational initiatives, technology transfer activities, and community outreach. Prior to her appointment in 2000 as NIEHS and NTP director in 2009, Dr. Birnbaum spent 19 years at the United States Environmental Protection Agency as a board certified toxicologist, where she directed the largest division at the EPA focused on environmental health research. Before that, she started her career with 10 years at NIEHS, first as a senior staff fellow in the National Toxicology Program, then as a principal investigator and research microbiologist, and a group leader in the Institute's chemical disposition group. Dr. Birnbaum is now a special volunteer at NIEHS and serves as a PI within its laboratory of toxicokinetics and toxicology. Dr. Birnbaum received many awards and recognitions. Linda, this is the part that I cut for time purposes. She was elected to the to the Institute of Medicine, now the National Academy of Medicine, because it got renamed in 2010, has also been elected to the Collegium Ramazzini, receiving an honorary Doctor of Science from the University of Rochester, Distinguished Alumna Award from the University of Illinois, among more than a score of other major honors. And words like hero, advocate, achievement, leadership populate those other 20 awards, 20 plus awards. Dr. Birnbaum has authored over 700 peer-reviewed publications, book chapters, and reports on pharmacokinetic behavior of environmental chemicals, mechanisms of action of toxicants, including endocrine disruption and linking of real-world exposures to health effects. In her 40 years of government service, she's also served on the faculties, adjunct faculties of the UNC Gilling School of Public Health, or Global Public Health, and the Integrated Toxicology and Environmental Health Program at Duke. We're pleased too to have appointed Dr. Birnbaum as an adjunct professor here at YSPH in our Department of Environmental Health Sciences, and she is playing a very active role. And if you had just been a fly on the wall when Vasilis walked into my office to tell me why he thought you should be a member of our faculty, and he had me at Linda Birnbaum, but he went on for a while, and I was happy to hear his thinking as to what you could do to help us in our vision of environmental health sciences. So few people in the field of public health have had such a profound impact on environmental influences on health and human disease, promoting Professor Winslow's goal of, quote, preventing disease and promoting health. It's an extraordinary honor and privilege for me to present the CEA Winslow Medal to our esteemed guest. If someone can actually show, there it is. There's where the medal is. I should have figured that out before I started. And so we'll now give Professor Birnbaum our school's highest honor, and we'll take a photo or two, and then she will present her lecture, Environmental Health, Past, Present, and Future.
I'm going to be a little relaxed as a backstage person.
Come on the projector.
Yes, on your face. Oh, the projector. They don't want the projector on our face. Let's all the way together. How's this? And I forgot to also acknowledge Dr. Birnbaum's husband and daughter Lisa. Lisa is a graduate of the Yale School of Drama. So there you go. I don't want that. Vasilis, could you join us for a moment?
Yeah.
Is Melinda Irwin in the audience? She may have had a conflict. Okay, that's fine. You've got it. Thank you.
Oh, brilliant. Somebody figured this out. I think probably there we are. Brilliant. Thank you.
So thank you all for being here, and thank you everyone for this incredible honor with this really special medal that I will cherish. Why did I close the box? I don't know. Oh well, we'll leave it open for now. It's a challenge. I think that Professor Winslow understood that prevention is better than a cure. If we can prevent disease, how much better is that than trying to treat and cure? And I would say that in much of my career, and certainly during my almost 11 years as director of NIEHS, that was a focus of what we talked about, was let's find ways to prevent disease. It may be easier to change things in our environment than it is to change our genes. Now, people might say that's changing. We have CRISPR-Cas9 and we've got other systems. Still not easy, and that's still on an individual basis. And with the opportunities that public health can afford for major population advantages by changing policies, I think we need to focus on that. So what I'm very briefly and very quickly going to do today is kind of give you a little snapshot view of where we stand with environmental health, first getting a little bit about the past. And these are just little kind of vignettes I pulled out. I mean, I think we've all heard how the Romans eventually, were defeated because of the tremendous amount of lead that was present in their water and in their cookware, for example. Whether that's true or not, I don't think we really know, but that's one hypothesis. And I hadn't realized before I started pulling this together, and I found this great quote from Ben Franklin about the importance of lead, that he concluded that occupationally related colic in a group of typesetters was caused by lead. I don't think that's something that's generally recognized. Then, of course, we have to see about John Snow. And when I think anyone in public health knows about taking the pump, taking the handle off the pump in London, which basically helped to end and prevent future cholera epidemics. I think these are standard understandings. And then these are just some nice pictures, I would say, of air pollution. And I say nice, being very cynical there, but I think we're all aware of what has happened dramatically with air pollution, whether you all know about all of these. If you watch The Crown during the pandemic, in the first episode in The Crown is focused on what happened during the London fog, with the actual deaths that were occurring and the damage. And then Delhi is often considered the most highly polluted city in the world for air pollution. Sometimes other cities try to catch up, like Beijing or Accra, Ghana, or a number of other places. But air pollution is, when we were in Delhi in 2015, we literally could not see the Taj Mahal until we could almost touch it. And that wasn't in Delhi, though, that was in Accra. But anyway, and many of you may know about the Denora fog that occurred in Pennsylvania. It was an inversion, just like the London fog involved inversion as well. So, and then the modern environmental health movement really got started by Rachel Carson. And if you haven't read her book, it's not a hard read. And I think it's important to understand how she kind of, by careful observation, was able to understand. So now kind of a little tour, just things I pulled out. Up in 67, we've known, my goodness, that is almost 55 years ago, or it is, that asbestos was linked to mesothelioma, and EPA is finally moving to regulate asbestos. It has not been regulated before, which is absolutely incredible. The problems with lead, Herb Needleman, if you don't know his story, what he first observed and reported on it, I mean, he was so roundly, awfully attacked, literally by industry, and not believing that, in fact, these impacts could be happening with lead. Really, some of it wasn't all the high level of exposure. Well, of course it was high. I think many of you may know that one of the most effective public health measures was removing lead from gasoline in 1976. And the level in American blood dropped to 15 micrograms per deciliter, which is way high compared to what is recommended today. The linkage of DES to cancer by an observant physician, where he was finding teenage girls and women in their young 20s developing vaginal adenocarcinoma. Now, it's not really an environmental, except, as you'll see in a few minutes, I define environment as including things like drugs that we're exposed to. And then the linkage of secondhand smoke to cancer, again, about 40 years ago. Then I think the first genetically modified mouse was formed in 1989, and that has really revolutionized a lot of not only testing, but mechanistic kind of approaches. Back to air pollution, the Harvard 6 city studies, which has grown to the 8 city and the 12 city and the 24 city, and the observations about how air pollution increases morbidity and mortality has been repeated in numerous countries around the world. I can tell you that, let's just say the polluting industry continues to protest these and try to fight the use of this original study in regulatory decision-making. BRCA was one of the first cancer genes that was actually cloned and has an increased association with breast cancer, as well as several other kinds of cancers. And the prostate cancer suppressor was closed. So you not only had genes that were in essentially, BRCA is a DNA repair gene, and that's actually involved in kind of pushing cancer. And then if you lose some of the suppressor genes, that also has an impact on cancer. PCBs, they've been around. They were first synthesized and they were considered wonderful occupational chemicals, first made in 1929. They were actually the first thing, one of the few things that EPA has actually banned, and they were banned in 1977 when TSCA went into effect, one of the only couple things that was banned. But the fact that PCBs not only are carcinogenic and reproductive and developmental toxicants, but the neurotoxicity became clear. Methylmercury in seafood was linked to developmental delays. I'm not talking about only the high levels of exposure that occurred in Japan, leading to Minamata disease, where you had actual mortality as well as severe morbidity being occurred. Lead keeps coming back to welcome us. So it was found in breast milk. You know, why should we have women have to be concerned about how the best way to feed their baby by the contaminants in breast milk, and lead was one of the first ones to be shown. And then the fact that we could show clearly a product of a mold, aflatoxin causes liver cancer. So, and I was talking to some of the students this morning, and I mentioned that, you know, 25 years ago, we thought liver cancer was not a problem in the US. And I'll come back later. It's a major problem now, and it's really related to the dramatic increase in obesity, much of it. Coming into this century, air pollution was linked to all-cause mortality. Initially, I think we thought that air pollution was just pulmonary and then cardiovascular, but now we know air pollution does, you name an organ system or a tissue, and air pollution impacts it. I think the understandings with Alzheimer's, we still have a long way to go. There are associations, by the way, between air pollution and also increased risk of Alzheimer's disease, as well as several other neurological problems. We think about allergies and asthma, and I think many of us think about dog and cat allergies. We think about the hygiene hypothesis and what, you know, how that may prevent allergies by early exposure. But I would say in many of our inner cities and poor communities, we have real problems with dust mites, which are associated with increased risk of asthma as well. And the last one here that I have here is kind of a little, I don't mean it to be cynical, but it's true. We focus so much on the impact of the mothers, on the developing fetus, and on the baby, and often we forget that fathers play a major role too, and there is growing evidence looking at preconception as a window of susceptibility as well. Back to air pollution, ozone is associated with an increased risk in asthma, and I can tell you, visiting communities in Long Beach, California, Long Beach and LA, which have 60% of all the goods in the US come in through those ports. And there are schools and there are homes right by the shipping areas. And the kids, when they play outside, the level of asthma in those kids is phenomenal. And a nurse at the school said, you know, we put in an air filter in the schools, and that had a major effect on helping the kids. Many of you may remember the anthrax episode in 2002, where several people received letters that contained anthrax in them, and I think 1 or 2 people actually died. But that understanding its mechanism was important. Again, back to lead, lowering IQ at 10 micrograms per deciliter. And I'll just go ahead and let you know, if you probably all do, but the CDC has concluded that there is no safe level of lead, and continues now and finally dropped the level from 5, which was set in 2012, down to 3.5. And that basically 2.5% of children in America exceed that 3.5 microgram per deciliter level. We began to look at the epigenome. Now, this is not the same thing as Lysenko, but the point is that the epigenome, we're understanding that it's extremely important. That's what turns our genes on and off, not only during development. Looking at cadmium affecting DNA repair, glucocorticoids and TNF alpha and their role in inflammation, many other roles for that. I think the point of benzene, I would say 30 or 40 years ago, when I was doing some pharmacokinetic and exposure work with benzene, we thought 10 ppm was a perfectly safe level. Well, we now know that you can detect sister chromatid exchanges and chromatid aberrations in people who are exposed occupationally to 1 ppm of benzene. So as you develop better techniques, you get lower levels. I think the point of artificial light, I think many of you are aware of the fact that shift workers, women especially, are at increased risk of breast cancer. And they're because we're interfering with circadian rhythmicity. ER beta, I think it was about 1996 when it became clear there wasn't only one ER receptor for estrogen, but in fact, there were multiple ER receptors. And ER beta is important in granulosal cells, which are part of the ovary. OP pesticides, which are still in use all over at high level. There's differential metabolism of these, and so that can impact the people's susceptibility. Hexavalent chromium, which EPA first evaluated with a iris assessment in prior to 2007. They are still looking at it 15 years later. but it clearly causes cancer in experimental animals, and there is some human data as well. And then we began to move into the, let's say, into the new approach methodologies, the NAMs using and that was my robot in at NIH, not mine, but at the NTP robot. BPA, you've probably all heard of BPA. That was eventually banned. FDA actually banned it from sippy cups and baby bottles in 2012. But guess what they've done? And this is a whole problem of unfortunate substitution. They moved from BPA to BPS, BPAF, BPA, etc. And when you start studying them, you find they do exactly what BPA does as well. Increased use of personal sensors. Those are actually look like some microarrays and then some others, and I'm going to come back to that later. I'm going to come back more to the whole issue of the exposome and the research there. And I'm going to come back to the importance of climate change. But one of the first things I did as NIEHS director is I put together a major review of what was known about climate change and health impacts that we published in 2010. So the Deepwater Horizon explosion, in which 11 people were killed, but people don't remember that as well as the fact that this was the largest oil spill that has occurred anywhere in the world, and really prompted the need for being able to know how do you conduct research in a disaster situation, which led to a whole development of a program that NIH, cross NIH now uses and is also being adopted by parts of CDC and so on. Arsenic, this was mechanistic. We've all, not everybody, but many people know that arsenic causes cancer. There's clear evidence for lung cancer, for inhalation, for kidney cancer and bladder cancer and skin cancer, but at least it's clear that some of it involves the recruitment of cancer stem cells by the thing. Flame retardants, there's a huge number of flame retardants, and this one class, this is not one flame retardant, this is a class of potentially about 209 different chemicals, and that causes cancer in rats and mice. That was some work that we did at NTP. And then back to epigenetics and actually epigenome, and NIH had a major effort in mapping the epigenome. So here we're getting back to kind of Professor Winslow's definition that environmental health is determined on physical, biological, social, and psychological factors. It addresses all human health related aspects of the natural environment and the built environment. And the important thing, it's not just say pollution, it's the quality of air, water, and land, but it also includes noise, housing, waste, physical factors, and vector control. And I think I might have a few more when I come back to this idea. Oops, and many of you have seen this picture, but I can remember as a kid in the 1950s when there were trucks go through a neighborhood spraying DDT to keep down the mosquitoes. And it was a great thing for kids to have lots of fun playing. So health and the environment actually have interacting and reciprocal effects. You have chemical pollution of the food chain. So, you know, we think about persistent organic pollutants and heavy metals, and who are the most highly impacted by these? They're actually the people in the far north, the indigenous hunters and fishers, whether it's in Alaska, northern Canada, whether it's in, for example, Greenland or the Scandinavian nations. Those are the most, because they are eating at the top of the food chain, and that is their traditional diet. Now, we don't want them to start having McDonald's all the time. That's not a healthy solution, but this is kind of the tension. We've had, for example, if you degrade our agricultural lands, which we know has occurred in many parts of the world, you end up with less food, which is a problem. If you degrade the water, I used to think that the major problem of the 21st century would be scarcity of clean water. I mean, now I think we all agree that it's climate change, but that is also related to the issues of clean and safe water. Air pollution is driven largely by the use of fossil fuels and petroleum products. So every time you see something telling you how we've got to have clean gas, clean natural gas, stop and think about what that is doing to our environment. Yes, it's better than coal, but problematic. We have a huge loss of biodiversity going on, and many people are very happy if there are fewer insects, but that has impacts because we have integrated systems, and if maybe you're not going to have pollination occurring. think we're all aware of what's going on with the NIEHS and the decrease in fees, for example. And then climate change, and I'll come back to this later, but again, it's clearly associated with the use of fossil fuels, has huge impacts on humans and wildlife, and also leads to an increase in natural disasters because of the droughts and the floods. Now, we also live in a very divided world. We have an increasing gap between the rich and the poor. Not only certainly within our country, but it's true around the world. In poorer countries, there is less access to clean water and sanitation. There is higher morbidity and mortality and higher injury rates. And then what's happening in part as a result of climate change, we're having even greater urbanization. We're going to have many climate migrants where our population growth may actually be outstripping our resources. And we have continued pollution of our rivers and oceans going forward. So if we look at the environmental burden of disease, what you can see is that the countries which make more money, the richer countries, where the gross national income per capita, for example, the US is all the way on the right there. That means they're also the ones with the lowest burden of disease. I think it's interesting when you look at it, not only, for example, are India and Bangladesh among those with the smallest GNI, but also the highest dailies. But if look at where Russia is, and some of you may remember that when the Soviet Union fell in 1991, within a year or two, the average life expectancy for men in Russia dropped to 55 years. And part of that was due to alcoholism and suicide. But I think that Russia is, and I think many of us are hoping we'll get in more trouble right now because of what's going on politically. But I think that it's important to look at it this way. And then if we actually kind of try to quantitate the environmental burden of disease, we know that essentially a quarter of all deaths are related to stressors in our environment, close to 13 million deaths a year. Much are caused by non-communicable diseases. Now, when I say that, I'm ignoring what has happened in the last 2 years and several months with the pandemic. I'll come back to the pandemic at the end. But barring that, I think we've done a marvelous job at controlling infectious disease in many populations. The environmental risk factors are contributing to at least 100 diseases and injury types. And that, this is the one where it says 1.7 million children, but I think there's even evidence the numbers may be higher related to the number of children that could really be saved with healthier environments. Indoor air pollution, which I'll reference again later, is a major cause of illness and death in children under the age of 1. And I think something we need to keep reminding ourselves are complex diseases have complex causes. You know, the simple relationship between a single gene and a disease is very, very, very rare. And even between a single environmental contaminant and a disease is rare. So and again, this is where forget the pandemic for a minute, but the global health trend has really been that we're heading for an epidemic of non-communicable diseases. And examples of that are things like type 2 diabetes and obesity and heart disease and cancer, asthma and autism. I was just talking with the students this morning about how the prevalence of autism in 8-year-olds has increased in the past 20 years from 1 in 120 children to 1 in 44. That's way too quick to reflect changes in our genes. I think if you look at it, if you look at both at the bottom one on your left, non-communicable diseases, you can see that the line is going up while the communicable diseases are overall going down. And if we look at the deaths, it doesn't matter. While the high-income countries are reaching that point of kind of most diseases being related to non-communicable, non-communicable diseases, that's what's happening even in low-income countries. We're moving in that direction. So this is just one example of the increase in incidence of these non-communicable diseases. This is some data from CDC, and that was published in the New England Journal of Medicine. But if what you can see is the color code change from 1965 up to 2010 in the incidence of of obesity. And actually, you could overlay this with type 2 diabetes. the incidence is very similar. But if there's actually, and I really should put the 2020 data is now available. When I last saw some data, the only state in the country that wasn't, for example, that dark red or orange was Colorado. Sorry, Gassets. But anyway, and if you look,
Top one is Greece.
with the most obesity,
Unfortunately.
right, which is interesting, and the US is about number 5 there. And I think this is something and not all obesity is due to being a couch potato, not exercising and what you're eating. There are compounds in our environment which predispose to obesity, or maybe difficulty in losing weight. Both appear true. So what is our environment? I think this is the pretty slide you might have been talking about, but our environment involves not just, for example, synthetic chemicals, the agricultural chemicals, and the and pesticides, not just personal care products, not just, for example, prescription drugs, but for example, our microbiome, which we are beginning to understand is a major contributor to our health as well as disease, weather extremes, stress, food and lifestyle, infectious agents, and so on. So it's much more complicated than people just thinking about pollutants. And always, and I think, Stan, you mentioned this, that the genes and the environment, it's always the interaction between the two. Certainly that pendulum is going to be closer to genes for certain kind of health conditions. Think of PKU. If you give the baby, avoid any phenylalanine, that baby will grow up to be mentally okay. But if you don't, so there's a gene and environment interaction there. And other things are going to be largely the environment that are going to change. And we just need to remember that. So this is just some estimates that came out from the Lancet a few years ago, but the global estimated deaths, and these come, from WHO and IHME, and I think everybody keeps hearing IHME because they're the ones who keep, along with Johns Hopkins, keep modeling what's going on with the pandemic. But the environmental chemical exposures cost more than 10% of the gross national products globally, which I think is important. And what you can see is that air pollution is the largest driver of this. I would say that's probably true. We don't know nearly as much about the increased risk of mortality and morbidity. And I'm not talking infectious agents here, but from pollutants in water or soil, chemicals and metals. I mean, it just hasn't been looked at nearly as well as air pollution. So we should think about that. And again, there's my one of my taglines, that the good news is much pollution can be eliminated and pollution prevention is cost effective. So health effects of pollution, these are just some wide number of things that can impact, for example, headache and fatigue, respiratory illness, gastroenteritis, it could be bacteria, it could be microbial, it can be chemicals. Certainly we know lots of things can impact our brains and our nervous system, certainly all, not only things in air pollution, but things, for example, like volatile organics, many of which we have in our household products, the increased cancer risk, pancreas, pesticides, and so on. But I think some of the key focus today for environmental health is focusing across the lifespan. I think much of the early environmental health work came out of occupational health, where you were largely focusing on adult men. And I think what we realized is that, first of all, sex matters, and males and females often respond differently, and that there are different times in our life when we have differential susceptibility. We're focusing on chronic and complex diseases. We're trying to change exposure science. I'll give you some examples of that. We are finally, not very successfully, trying to combine environmental exposures. No one is exposed to one thing at a time. We all live in a soup, not only of chemicals, but of microbes and noise and physical factors and so on. We have emerging environmental threats, things we didn't know about. And I can mention some of them later. We have the whole major issues of environmental justice and health disparities, which have major impacts on how we people respond to different kinds of stressors. And I can tell you that can all be modeled in laboratory animals as well. some degree. We're focusing on the global issues because we have a very small world, and if the pandemic hasn't taught us anything, it should have taught us that the world is tiny. We're focusing on epigenetics and stem cells, the exposome, the microbiome I've mentioned. We're trying to develop predictive methods, and we are clearly engaging communities. And I think one of the most important things is we are trying to figure out how to use all the computational power that exists today and the huge amounts of data that are being generated. So artificial intelligence is already starting to be very helpful as we go forward. And I think the really important thing is translation in a public health is not necessarily bench to bedside, it's bench to bedside to community. So some of the things that have been very important in moving us to where we are today have related to environmental issues, and toxicology really are some of these National Academy efforts that we sponsored along with EPA, looking at tox testing in the 21st century, which is moving towards alternative methodologies, exposure science, and the whole using 21st century to improve risk-related evaluations. We've got to stop focusing so much on some of the approved guideline studies that were developed 50 years ago. They often are not asking some of the questions that we need to ask today. So there's conceptual shifts that have occurred, moving from the old where chemicals act by overwhelming the body's defense to where chemicals act by disrupting control of the development. And while the acute toxic forces were toxicity was often at very high doses, these developmental effects and the chronic effects are very low dose exposures. We have the immediate cause and effect based on studying adults, and then the long latencies following developmental exposure. We used to focus on adults, as I said, and now we're focusing on children and the developing fetus that are often most at risk. And hopefully we're moving from one chemical at a time to reality as always mixtures. And this involves the use of multidisciplinary teams. I don't think you can do environmental health today without assembling groups of people from different disciplines. I'm just going to get a drink of water for a minute. So this is just a list of some of my favorite chemicals. These are some of the ones that today we know are the bad actors. And what you can see is the first one I have up there is BPA. We know BPA is bad. Industry one tells you it's fine. You may remember that it was initially made as a estrogen, synthetic estrogen, but DES was much more effective. So that was the end. So then they began to use it extensively. And again, you know, you may have bought your new water bottles that say BPA free. We've got the other things in it. We've got the phthalates, and phthalates, the more we study them, the more we learn. They can be carcinogenic. We know that they affect the developing male reproductive system. There's also evidence that they impact the developing female reproductive system, and they are clearly developmental neurotoxicants, among other things. PBDs, I mentioned them before, those are kind of flame retardants, which are no longer being commercially made. But guess what? We all have them in our bodies anyway, because it takes a long time for them to be eliminated. PCBs, as I said, they stopped being made in the US in '97. The last country in the world to intentionally make them was the Czech Republic, and they stopped in 1995. But the point is, everyone in this room has them in their bodies because they keep getting re-entrained in the environment. My favorite is dioxin or TCDD, never intentionally made, but an unwanted contaminant, which is associated with a whole host of adverse health impacts. DDT, that was the thing that Rachel Carson wrote a lot about. And remember, that was banned not because of its human health impacts, but because of eggshell thinning in birds. And that was banned by an act of Congress in 1972, within 6 months from when it was first proposed. And I should tell you that there's growing evidence that DDT and its metabolites are chemical obesogens, as well as associations, say, with type 2 diabetes. Lead, we've spoken about before, and everybody pretty much is aware of lead's impact on cognitive development in children. Many people are not aware that as many as 400,000 Americans die from cardiovascular disease every year, which may at least in part be associated with lead that they're exposed to. So people my age, when we were kids, we had a lot more lead than, say, children are today. But we know some are still being heavily exposed. Mercury, a lot of mercury in the environment is from the burning of coal. But there's also mercury, for example, in artisanal gold mining. So in both in Africa and in Latin America, mining of gold is often done under very, in very poor communities. And the using mercury to help isolate the gold has led to mercury getting into the aquatic food chain and getting into fish. And then the people are eating the fish contaminated with methyl mercury, and we're seeing adverse effects on the children. Arsenic, I mentioned before. If you have a young baby, don't exclusively, when you first stop nursing, don't exclusively give it rice-based products, because a lot of rice is contaminated with arsenic. There's an active transport system in the roots of the rice plants that takes up arsenic, and that can actually lead to elevated arsenic. Most of us have no idea where our rice is grown, and many parts of the US and other parts of the world, this is a real problem. So I'm not saying don't ever give your baby rice cereal, but don't make that their major food. Anyway, and then PFAS, and I'm happy to answer questions about this favorite. I don't think people realize, I didn't realize that PFAS were first began to be used commercially in the 1950s. And basically they flew under the radar with heavy use until close to 2000. And in the last 20 some years, a tremendous amount has been known, but there's a tremendous amount more to learn, because there's not one PFAS or two PFAS or three PFAS. On EPA's website, they now list 12,039 different PFAS. So you tell me how you're going to test every one of those. It's a whole nother discussion. And there was plastics are something that we really have to be dealing with. Not only because of what is absorbed to the plastics or used in making the plastics, but we're now finding microplastic particles in serum. And I think this was just an article from the Washington Post a little over a week ago. So again, air pollution, to come back to that again, is a complex mixture with a wide variety of adverse health effects. And remember, air pollution is not just the NIEHS pollutants, which are lead, carbon monoxide, sulfur oxides, nitrous oxides, PM, and ozone, but it's also the VOCs as well. And I should say that half of the VOCs that are in the outdoor air are coming from indoors. So think about what your indoor air is like. So household air pollution, again, here in the US, we tend to think about it as a problem of less developed countries, but let me tell you, there are places in our country where it is a problem as well. So for example, in some rural areas where people are cooking, for example, with wood stoves, or in some of our inner cities where there are small apartments which are heated, and both heating and cooking are done with gas, the levels of indoor pollution can be very high. In fact, usually the levels of indoor pollution are much higher than levels of outdoor pollution. And yet indoor pollution is not regulated. And that's why there's been, I think, much less work done on it. And I think there was an interesting paper that some of the epidemiologists at NIEHS did, showing that indoor wood burning even once a week was associated with a higher risk of breast cancer for women. So now the endocrine system. The dean mentioned endocrine disruption was something I've been very involved with since it was first kind of identified as a potential result of environmental chemicals. But the endocrine system, I think, as most of you probably know, is an extremely complex interacting system where you have fine-tuned effects. It's very sensitive to perturbations and naturally operates at low dose. I think one thing is there are differences when you're talking about the endocrine system during development, where the effects are called activational and are actually often not reversible, as compared to adults, where the endocrine system may be modulated and you may be able to reverse the effects. But the World Health Organization in 2012 decreed that endocrine disrupting chemicals are a global threat that needs to be addressed. So we have the whole issue of lots of diseases that are associated by developmental EDC exposures. And again, if you look at this with some of the chemicals I pulled out that cause them, you see some of my favorites, cancer with BPA, endometriosis with dioxins and PCBs, infertility. I mentioned phthalates before, but also pesticides, diabetes, BPA and PCBs, early puberty, obesity. I know I have PFAS in here somewhere. Maybe I don't, I should. Well, I do. Perfluorinated, it should be poly and perfluorinated compounds with increased susceptibility to not only suppression of the immune system, but also increase in autoimmunity as well. And again, a whole variety, almost all of our systems have an endocrine involved and the endocrine system at some time. And there are many different kinds of exposures which can perturb that endocrine system. So I think what we need to understand with endocrine disruption, there's still a lot of pushback on this whole idea. And I hate to remind people that people take endocrine active drugs all the time. And why do we think sometimes that synthetic chemicals or naturally occurring chemicals behave any differently than things that are intended to cause a perturbation? But we know there are many agricultural chemicals that are actually endocrine disruptors, things like atrazine, for example, which is banned in most countries in the world, just not here in the US. Many food additives and packaging materials. I have to tell you, plastics are loaded with all kinds of endocrine disrupting chemicals, whether it's phthalates, or BPAs, or PFAS, or some of the other bisphenols, or things like parabens. It's all there. So please, nobody heat their food in plastic, which during the pandemic has sometimes been a challenge, because you would get takeout, food, and they would come in these nice plastic, and you're tempted to just stick that in the microwave. Don't. So again, many industrial chemicals and byproducts, PCBs, for example, disturb many endocrine systems. Waste products, again, the dioxins, the PAHs, and I just put this the bioaccumulation and or persistent related to some of these chemicals. You have your pharmaceutical products and phytochemicals, things like zearalenone, for example. And then you have personal care products. And I was very upset within the last year when it came out that makeup was loaded with PFAS, various kinds of makeup. PFAS are everywhere, but we also know that you have BPA and phthalates and so on in personal care products and with coatings. So low dose, and I'm going to go to the bottom of the slide in a second and say, what do we mean by low dose? By low dose, do we mean lower than what? Lower than a high dose? Well, what do we mean by a high dose? I define low dose as an environmentally relevant dose. And I would remind you that if you're an experimental scientist and you're working in the lab with little rats and mice, you are going to have to give a higher administered dose in order to achieve the same internal concentration as with a big human. And so if you see studies that are done what appears to be a ridiculously high dose in animals, you really should ask the question, but what does that mean internally? What is the serum concentration in those animals, and see how that relates to what we know in people. So again, when I look at the literature, and when I know a lot of the work that I've done, we focused on trying to match serum concentration. So the work where we show that for dioxins or PCBs, it was what we call the body burdens. That was an appropriate metric to compare to humans, even though the delivered dose was about 30 times higher a day to get it. Arsenic, you've got to give about 10 times higher to your animals than people would, say, be drinking it. And again, we are exposed to lots of EDCs as well as other chemicals. So I've mentioned the whole windows of susceptibility concept before, and that is that, what occurs early in life can impact you at various stages of your life. And this should really be a much more complicated picture, because our windows of susceptibility are not just in gestation. They're also, as I mentioned before, preconception. There's some evidence, especially for stress there. Childhood is a window of susceptibility anytime the body is rapidly changing, growing, cells are differentiating. So pregnancy is not only the fetus that's at increased risk, it's also the mother that's at increased risk. And then when you get into later years in life, those can also be windows of susceptibility. And what we've said a couple of times is that early life exposures have lasting effects. And this is the whole developmental origin of health and disease. It's led to a whole new really field of research and organizations called the DOHAD approach. But again, when the organs are forming, when gene expression is being established, key role of epigenetics during development, and again, changes occurring are permanently altered an organism. And many people give David Barker the credit for the development of this with his studies where he was studying the hunger winter in Hungary that occurred in 1944, and grandchildren of the people who were basically had minimal nutrition during that have increased risk of cancer, heart disease, type 2 diabetes, and so on. So again, the fact that exposures can impact several generations down the road is something that has to be looked at. And I think what this has led to more questions of people saying, are we exposing our animals at the wrong time? If we're doing all our studies in adult animals, when in fact this critical window is developmental, we should look at that. And we need to look at, well, when should we be measuring exposure? I have to tell you, it always drove me crazy with the Long Island breast cancer study, where they were busy measuring the chemicals in women who had breast cancer. Breast cancer doesn't occur overnight. It occurs over 10, 20, 30-year time frame. You really need to know. So the importance of prospective longitudinal studies in our human population cannot be, I think, overemphasized. And we have the key exposure questions. Is it acute or chronic exposure? The developmental windows. What about the past histories? Mixtures being the reality. And what do we really measure? Are we measuring a chemical specifically, or can we look at responses as an indicator, a biomarker of effects? So I've mentioned mixtures a couple of times, and part of that is my frustration and having been doing this work for over 40 years, and people are still doing single chemical exposures rather than more complex kinds of things. And you can work with defined mixtures, and this has been done with, like, say, 2 to 50 different compounds. You can look at source emissions. You can look at multiple sources, but there's tremendous complexity in dealing with the real world scenario where you have both the chemical and the non-chemical stressors with multiple routes of exposure. And somehow we need to begin to approach that. And the approach involves something called the Exposo, which is defined by Chris Wilde as the totality of exposure from preconception to death. Obviously, that's almost impossible to measure. In fact, it is impossible, but you can measure as much as you can, even if not the totality. You can integrate at multiple scales, and you can really use data-driven approaches to identify stressors that impact health. And by that, I mean some of the untargeted approaches, which are so important. Otherwise, if you're only looking for things that you already know might be there, you're looking under the lamppost, because maybe it's the things you're not looking for that are really important. So the complexity of the exposome is the stressor, the source, the place, the time, the route of contact, the distribution, and the targets. And we have all kinds of great new technologies. I used to like the one, I don't have that one up here, but there's actually a little camera that's been developed if you want to measure dietary exposure. People are notoriously bad at telling you what they ate yesterday or how much they ate yesterday. But if they actually took a picture every time they ate something, that has a measure, some ability to measure the size, that can be much more helpful. We have all the different kinds of sensors. Many of us are wearing, Fitbits or Apple Watches or something where you can actually get some biomedical measurements made. You can also get measures of exposure made at all, certainly of physical activity. And there are ways that you can be monitoring for a whole range of compounds. And then there are biological response indicators where you may be looking for changes in, for example, gene expression, or you may be looking for changes in your metabolome, or maybe your lipidome or your proteome. And so there are many new integrated tools and technologies for exposome assessment. Questionnaires are still extremely relevant and extremely important. Lots of work is being done with all the GIS modeling where you can look not only at air pollution, but you can look at issues of water pollution or water systems. You can look at issues of soil pollution, and you can begin to get some ideas of what the exposures are. I've mentioned some of the mobile devices. One of my favorites are the wristbands. And the only problem that we had with our children's studies that we were using wristbands is they didn't want to give them back because they were these bright colored bracelets and they thought they were fun. Well, we could give them a new one without all the sensors in it. That would usually solve the problem. But we're really dealing with lots of knowns, known knowns, known unknowns, and unknowns, unknowns. And that, again, the pictures I mentioned, and we do the different biomarkers, and then we can do the omics technologies. And there are high throughput methods that are being developed for that: epigenomics, transcriptomics, proteomics, metabolomics, lipidomics, glycomics, even more. So, and again, we can really have to be using the targeted, both targeted and untargeted analyses as we go forward. We are doing more and more requiring or encouraging systematic reviews where you have an a priori process that defines what you're going to look for and what are the quality of what you're going to require. I would remind you that systematic reviews still require scientific judgment. So you may find two groups do systematic reviews and one group throws out lots of things because it doesn't meet one of their predetermined criteria. The other group says those are perfectly relevant and continues to use them. So be a little skeptical when you look at some systematic reviews to see what they have not looked at or avoided. The new approach methodologies is a huge approach because we've got too many chemicals and not enough data. For PFAS with 12,039 chemicals now, I'm sure it'll be more next year. You still can't test even with new approach methodologies, that number. But using computational approaches, and I think what we're moving more is to trying to understand the targets or pathways which are linked to toxicity or adverse outcomes. And we're developing lots of high throughput and in vitro and computational approaches. So there are the scientific and non-scientific, and I'm almost done, guys, challenges of NAMs. We have our animal methods are currently the reference for human. Some people will tell you animals are not good models for humans. I'd remind you all that people are animals. And in many cases, if you see effects in multiple animal species, it's likely that at least some humans may be susceptible at least some level of exposure. But using some of the NAMs, trying to cover the whole chemical space, there are issues with the availability of alternatives, and huge issue is how do we get the regulatory agencies to accept them? Another approach that's being used is key characteristics, and I'm sure everyone in this audience recognizes who that is on your left-hand side. That's former President Obama. But the key characteristics admits we can't test everything. So we're trying to prioritize chemicals for assessment, and we're looking at things. Key characteristics is really another term for pattern recognition. And if we can recognize what some of the key characteristics are of chemicals that are biologically active as carcinogens or neurotoxins or immunotoxins or hepatotoxins, I could go on, we can develop a more holistic view of how to approach chemicals. And I think this is really an exciting approach. It's actually being used certainly clearly for hazard identification. For example, by California, Cali, PA, and for example, by the World Health Organization. So as we go forward, I think we have to understand that the role and importance of mechanistic understanding is only going to increase. There are going to be fewer animal studies, traditional ones that are done. Mechanistic information is going to have a bigger role, and some of it's going to come from in vitro or short-term kinds of studies. And there's going to be more mining and data analysis from repositories. And I think key characteristics are maybe an effective way to help organize information. But we're still going to need expert judgment. So always look at where that's coming from. Now, just to remind you, oh, come on, start moving. This is one of my favorite slides. This is showing you the global temperature change and how it is continuing to increase. And if I stand here long enough, we'll get to 2000 and I think it's 2016 or 17. And what you can see is, as we all know, there's no way we're going to avoid the 1.5 degrees C. In fact, we're almost at it right now. And whether we can avoid going over 2 degrees C is still known. There's been a tremendous increase with weather and climate disasters. Many of them are being driven by climate change. So the increase in floods, the increase in forest fires, the increase in droughts. This was the 2017. These are all the episodes that were more than $1 billion. And the numbers in 2020 are much higher than numbers were in 2017. There have been more climate-related disasters, whether it's hurricanes or, for example, flooding and so on. So climate change and health, our exposure pathways may differ. I think that's something to think about. So we know that some of the things we think about are heat waves, air pollution, food security and nutrition, our vectors, hurricanes and flooding and drought, and there's a whole range of health effects. So every time somebody talks to you about climate change and reminds you what's happening to the polar bears, please stress them it's also happening to us. Increased risk of mortality and morbidity, certainly increased mental stress on people. And so climate change is having observable effects on our health right now. They're associated with other social and environmental determinants. And if we could reduce, with the IPCC report that came out just a week ago, it's clear that if we can act, we can prevent some of the most serious changes, but not if we don't act. And clearly the health benefits outweigh the costs of meeting these goals. We're also outside the safe operating space of the planetary boundary. This paper came out in ES&T just recently for additional chemicals. In other words, the levels of pollution are already causing us huge problems. And just to remind us about COVID, where do we see who's at risk for COVID? Well, certainly somebody my age, if you've got chronic lung disease or you're immunocompromised and so on. But we now have growing evidence of the environmental contributors to COVID susceptibility and severity. Air pollution, there's clear data that plays a role. The PFAS, there are over 4 papers that have now been published showing that the body, let's say your serum levels of PFAS are inversely associated. The more PFAS you have, the more likely you are to get COVID and the worse that it is likely to be. And then we just don't know about a lot of the others, but other things we know that modulate the immune system, you would expect that they would have an effect as well. And we ought to think about this so to help prepare for the next pandemic. And I always like to end quoting Pope Francis, and this was in his Laudato Si from 2015, where he said, climate change is a global problem with grave implications, environmental, social, economic, political, and for distribution of goods. It represents one of the primary challenges facing humanity in our day. And with that, thank you, and I hope we have a minute or two for questions.
All right. I think I have to negotiate for the folks who are online so they can ask questions. Can I recruit 2 folks on each line aisle? Thanks, Ralph. And if you have a question, please wait for the microphone so people can hear. And then I'll negotiate this and let you. Sharing.
No questions. I didn't answer everyone.
I'm sure there'll be a question, and I think we can receive questions through the chat online if somebody has a question.
Okay,
Those of you who are online.
so Nicole has.
Yes, Nicole has a question.
Thank you so much, Dr. Birnbaum, and congratulations on this honor. I really appreciated the historical tour through our environment, and it's hard though not to feel a little discouraged when you had Ben Franklin identifying lead as an issue, and then now we still have 2.5% of children lead poison, arsenic, asbestos, not yet regulated by the EPA. So I'm just wondering if you feel optimistic or can offer any insight on how we can tackle, fracking, microplastics, PFAS, and we're still, still haven't appropriately dealt with some of these classic pollutants.
Well, as I've often said, which probably shouldn't be quoted on, but that, we have the best government money can buy. And that is true on both sides of the aisle, and our policies are determined by that. So if you look at some of our environmental regulations in this country, even if you identify a major problem, it takes at least about 6 years for any regulation to go into effect because of some of the appropriate requirements for input from stakeholders. But TSCA, which is the act that controls chemical and commerce, has been exceedingly ineffective. It was updated in 2016 and called the Frank Lautenberg Safety Act. Unfortunately, he would be turning over in his grave if he saw what it has become. And again, there are efforts to make it work, but you know, in Europe at least, chemicals have to be tested more like drugs before they can go on the market. So that's a huge problem in this. So it's here, everything is presumed innocent until proven guilty. And that's backwards what we should have with chemicals. In other words, the chemicals should in some ways be regulated like drugs are, where they have to be tested. If you ask me, I'm a cockeyed optimist. So I think that there are opportunities and there may be opportunities for us to do better. You know, I think as we realize problems are occurring, but I think you have to be alert to how things can be captured. So there's an interesting article today in The Intercept. Sharon Lerner is a really good environmental reporter, who's been reporting on some of the problems within EPA, and she's also been reporting on PFAS. And what she talked about today was how Chemours, which is the manufacturer of Gen X, which was the replacement for PFOA, they are petitioning EPA not to go ahead with the regulation of Gen X because they need it to deal with climate change. Excuse me, cynical, but you'll see this again and again from certain companies, especially our fossil fuel and energy industries, which will also tell you like about, you know, clean gas. So I think we have to be just aware of that. I think that the way things happen in government is you generate outrage. You know, just saying you got a problem doesn't mean anything. Getting a mass movement is how. So if you look at Greta Thunberg and what she has done with climate change, although it hasn't really moved us here, but it is moving in other parts of the world. So if we can't deal with some of these problems, I dramatically worry about the 22nd century and what the world will be like at that point. Not terribly optimistic.
Hi, Doctor. I have a question about the slide of the bad actors. On the right, you had the outcomes, and some were highlighted in red, and I just wanted to know a little clarification on that.
I'd have to go back and remember, maybe those are the ones that we have good human data as well as animal data.
Okay, thank you.
I should say that I made my slides, I sent a PDF of the slides and they are available.
Well, that's
You should
lovely.
make them available.
Thank you, thank you. Why don't I just, oh, there's one back there, thank you.
Hi, Dr. Birnbaum. Thank you very much for this talk. I kind of like follow-up questions. We talk about policy. There's a lot of talk about regulating chemical as a class, such as the PFAS, and I just wanted to talk about how likely it would be, and also like how we define a class. Sometimes, you know, like Gen X would be look quite slightly differently in the chemical structure, right? It's currently in PFAS, but yeah, just get it.
Okay, so that's a great question. I should say regulating chemicals as a class is nothing new. Dioxins have been regulated as a class of compounds for over 20 years, for example. PAHs are regulated as a class. When you accept the fact that things aren't 100% accurate, you know, that it may not be exact, but it's good enough, you can do that. So PCBs are regulated as a class. I would say bisphenols should be regulated as a class, and it may be need to be larger than just bisphenols. There may be other compounds. As with PFAS, I'm one who has been pushing regulating PFAS as a class. Why? Because they are all environmentally persistent. None of them are going to break down in the environment. They don't exist naturally. They're all synthetic products. They all are highly mobile. So, you know, everyone in the world now has PFAS in their bodies, as well as all the animals and so on. We know that even for chemicals with a short half-life, over time, if the levels in the environment increase, plus if you're exposed at a low level every day, the chances of adverse effects start increasing. Is my desire to see PFAS regulated as a single class going to happen? Probably not. But we've got EPA to at least talk about subclasses. And the National Academies has actually said that regulating chemicals as classes or subclasses is scientifically justifiable. So I don't see how else we can do it. I mean, there are so many compounds for which there is no information other than maybe their structure. And again, that's a huge problem. All the non-targeted screening is partially necessary because nobody has standards for these things. So you can get to the point by very elegant mass spec approaches. You may eventually be able to get a good idea of what it is. You may, but how much easier if we could get from industry standards for these things that they're making.
Thanks, Victor. I'm wondering how we're going to avoid the unfortunate substitution problem, especially when we're looking for certain functionalities. Any chemical you introduce is likely going to have a similar effect to the previous one.
Well, that's a great question. And first of all, you require testing before you can introduce a new substance into the market. So you make sure it doesn't do what the one you're trying to avoid. But I would say when you move from one chemical, which you know is bad, to another you don't know anything about, and you can use it in your industrial processes without modification or major modification, why would we think the body would behave any differently? You know, it's just, there hasn't been the thought. Now, Paul Anastas here is focusing on the whole issues of green chemistry or sustainable chemistry. And I think we need to begin to support the use of approaches which kind of look, you know, and look at things before you use them. The other point I'd make is that so much of our focus is on, you know, we need to do this. We need this functionality, as opposed to saying, what's the problem we're trying to address? And maybe there's a totally different way that you can, say, achieve, make your flame resistant product, a totally different way that doesn't involve chemicals at all, or dangerous chemicals. So I think we have to look more broadly and focus on the solution approach, not the problems.
I wonder if the issue of the environmental health legacies of war is something that you've ever dealt with and have thoughts about. It's something that's come to the fore, obviously.
Well, and it's appropriate that it has. I mean, it's really been, it was a huge effort to get compensation to the ranch, well, to Vietnam veterans in 1991, when it was finally said, you know, if you had boots on the ground in Vietnam, then we presume that you had exposure to Agent Orange and dioxins. That hasn't worked out all the way we would like it. But right now I am chairing a working group at the VA, which is looking at the tens and tens of thousands of samples that they have, both biospecimens and questionnaire data and a lot of computational data, to look at what can we still learn from these stored specimens that we have from folks who served in Vietnam. We also are looking at over 50,000 blood specimens that come from the time of the Korean conflict, from 50 to 53. Those were used to look at, I believe, infectious disease prevalence in the population, but there's lots else that can be asked about those specimens as well. So yes, I mean, I think that there is growing evidence that exposure to burn pits was not good for health. And you know, if you stop and think, what are you getting out of burn pits? You would expect it would. I often say if it smells bad, it probably is bad. And my former son-in-law was, you know, in the Middle East during two tours of duty there. And the levels of exposure he talks about, that he was bivouacked right next to these burn pits. And he has long-term health consequences. So I think there is more attention that's being paid. I would say the current Secretary of the VA is very open to exploring concerns about the legacies of war.
Let me end with a quick and controversial question. First of all, on the positive side, research can be very helpful. Think about secondary smoke research and how it fundamentally changed policies. And you know, if you can stop people smoking in bars, you can stop people smoking anywhere. And that has been a shift in our lifetimes. But my controversial issue is on the theme of radiation. So with 3 Mile Island, Chernobyl, and Fukushima, there is a natural reluctance to embrace nuclear power. Also, the failure to operationalize something like Yucca Mountain, where we might have the prospect of storage of nuclear waste, is also another good reason to oppose nuclear power. So safety of nuclear power and the waste of nuclear power. The flip side, of course, you know what I'm going to say, is that this is a carbon neutral energy supply. And if it were done safely, and if we had a satisfactory approach to disposal of nuclear waste, it could be a lot faster than going solar around the world. Now, that's a controversial statement, and maybe that's not true because it takes a long time to regulate and build a nuclear plant. But in theory, countries like France have been running on nuclear for decades without any, if you will, accidents. What is your opinion on that?
I thought you were going to ask me about cell phones. And do not hold cell phones by your head, but aside, other than that, personally, I think that we should be reinvestigating the use of nuclear energy. I had kind of moved there before Fukushima, realized that the majority of deaths from Fukushima were not from the reactor, but were from the tsunami. And about 20,000 people drowned. That was the flood. You know, I will tell you, if you're in Japan, everybody, well, maybe not everybody, but lots of people talk about the Great East Japan earthquake, referring to that. And it has had a major impact on, I think, the psyches of people in Japan because of what happened. But the real problems of that with tsunami, I mean, that's what really killed. Now, clearly, nobody wants to have excess radiation just floating around, but you got to kind of go back and say, from fossil fuels and petroleum products, are these not worse, and not worse for our entire world than having some nuclear power? So I'm not an expert in nuclear power, but I do think it's something that, looking for clean fuel, we have to be investigating again.
Thank you very much, Dr. Birnbaum. Your talk was excellent, and I want to thank you. I want to thank your family, and I want to thank all participants today for participating in what we think of as a historic event. So thank you again.
Thank you.